Wow.
Hello, everyone. We will just wait a few seconds to allow people to join the event, and we will start the webinar. Okay, let's go. So, dear attendees, welcome to all of you. We are delighted that you are joining us today for this live webinar on how Tebubio can support you to accelerate your mRNA-based proof of concept. Through this session, we will be showing you our microscale mRNA production service, our capabilities to provide you with an mRNA sequence tailored to your needs, and also how, thanks to our technology, we can support you from R&D to GMP production. But first of all, let me introduce you the team who will be accompanying you through this live session. First, our speaker, Romain Goulet-Cordonnier, who is our Chief Scientific Officer.
Hello.
Célia Bosso-Lefebvre, who is our Project Manager in charge of the mRNA project, so our mRNA expert will answer your question. And I, Frédéric Samazin, I will be your host and moderator for this session. So during the webinar, we will cover a brief introduction of our company and contract research service lab, a detailed presentation, of course, of our microscale mRNA production service, and finally, a live Q&A session to answer all your questions. So during the talk, don't hesitate to put all your questions in the Q&A session, and we will answer all of them at the end of Romain's talk. So without further delay, I'd like to hand over to our Chief Scientific Officer, Romain Goulet.
So hi, everyone. Thanks for joining. I will start by introducing briefly Tebubio. So let's start. So to be able to facilitate life science research every day, Tebubio has 9 local offices in different European countries. They are represented on this map with flags. Thanks to its strong network and 70 years of experiment now, we can distribute many solutions as reagent or biological sample all over Europe. And since more than 20 years, our laboratory is growing with our headquarters next to Paris, allowing us to propose more than just product distribution. So thanks to this unique combination between delivery and lab project management, we can help you focusing your effort on what is important for you, your research and your projects. So as I said, the laboratory is growing since more than 20 years now. It is composed by scientific experts in different fields and a pool of polyvalent technicians that can realize diverse services in different fields. Today, we propose a range of services solution for your protein production and cell culture projects, biomarkers and bioassay analysis, and of course, molecular biology projects. This is mostly thanks to our knowledge in molecular biology and the feedback from the current market and our own customer that we have developed our own mRNA optimization and production platform called RNAvenir, positioning ourselves in mRNA mini-scale production. This platform allows you to screen and validate different mRNA construction, matching your project needs. So thanks to this platform, we can start from gene synthesis till GMP production of mRNA by validating with you every steps, your sequence optimization, the production, the screening, the delivery system, the expression of your targeted protein within different cells model, and its analysis. So today we will focus on the first step of this workflow, the optimization and the production of your targeted mRNA. So let's talk about mRNA technology and what Tebubio can propose. In the past 10 years, technological innovation have allowed mRNA to become a very promising tool for therapeutic application. The COVID-19 outbreak has demonstrated the effectiveness of mRNA vaccines against infectious disease, vaccines that are faster to develop, easier to produce, and pose fewer health risks than conventional approaches. What we call the transcriptome is now the future of therapeutic, more specific than genetic-based therapy and challenging protein-based solutions. Of course, today we will focus on mRNA, but you have to know that Tebubio can work on general RNA projects. The mRNA is not only used in infectious disease vaccine, but also as a potential therapeutics to cure cancer, for CAR T cells production as immunotherapy tools, for antibody or protein-based therapeutics, or also for antiviral technology. The non-coding RNA is also widely used for analysis and diagnostic techniques. For those different reasons and applications that I've mentioned, there is an increased amount of requests for mRNA production and custom services. Several CDMO have been created to produce large-scale mRNA and GMP-grade mRNA. These productions are often expensive, with a waiting list and an extended lead time from weeks to months. Knowing that, Tebubio has decided to focus on small-scale custom mRNA production with a control cost and a short lead time to match with screening and research-grade projects. It allows you, as a researcher, to test several molecules in small quantity, as it is usually done for screening protein-based therapy development. It takes only 5 days to produce small-scale mRNA starting from the DNA template, with a scale range of 100 microgram to 2 milligram. As you may know, the mRNA is composed by different key elements. The coding region for your targeted protein surrounded by non-coding sequences which influence the final mRNA stability and the translation efficiency. A capping structure based on a high methylation of the 5' extremity protects the mRNA from degradation within the cells, which prevents the immune system from detecting it as a non-self agent. Last but not least, a 3' poly-A tail increases the stability of the mRNA molecule, limiting its degradation. To give you a general overview of our workflow, I can say that with any DNA matrices related to your final application, your mRNA can be rapidly produced with lower cost and high yield by your production workflow. We start our interaction by discussing with you about your sequence of interest, optimizing it together with the solution we have to improve both transcription and final translation. Then We synthesized and cloned your selected sequence in a DNA template, also optimized or custom made for your project. Once the transcription is done, the capped mRNA is purified and its quality is checked by TEPstation. With this process, once we have statuate on your optimized sequence and receive the DNA, it will take 5 days for us to produce your mRNA. So now I will go step by step to explain what we can do and what are our main optimization solution. So starting by optimizing your sequence. Thanks to our expertise, we can optimize your sequences to maximize the efficiency of the transcription and final protein expression, specifically for your final vitro and vivo models. Some sequences, in particular those with high amount of G and C bases, can form secondary structure such as hairpins generated by base paired interaction. Those hairpins lead to a lower efficiency of the RNA polymerase, which can decrease the final RNA production or generate truncated RNA. This is why our codon optimization services will include a G and C reduction and a limitation in the secondary structure formation. We also have the possibility to modify the coding sequence of your mRNA to limit its final immunogenicity. The immunogenicity is a major problem for some drug-based mRNA. The non-modified exogenous mRNA activate the TLR receptors within the cells, leading to the release of pro-inflammatory cytokines, generating general inflammation in the patient or in your model, which reduce, of course, the mRNA traduction by increasing its early degradation. To overcome this problem, therapeutics mRNA might be modified by replacing some nucleotides by analogous nucleotides, which reduce the immunogenicity and increase the resistance to degradation process. As an example, all the uridines from the first approved mRNA vaccine against COVID has been replaced by N1-methyl pseudouridine. To limit the immune response and improve the protein production, codon optimization proposed by Tebubio include the U depletion. You can see here that we have several possibilities to modify the U as pseudouridine or even the N1-methyl pseudouridine that has been used in the first vaccine developed against COVID-19. It is important to know that the N1-methyl pseudouridine is the most common modification, allowing a high protein expression in many cell systems. This is what you can see here on the graph. The gray bars are the expression rate of the mRNA modified with these analogous nucleotides, and it shows the best results in many cell types. Once we have fully optimized your sequence of interest, our next step will be to work on your final vector. For mini-scale services, we can start with your own DNA template or build custom template matching your needs. We have the possibility to adapt the capping. One of our strengths is also to have developed our own pDNA with optimized UTR and poly-A tail, which improves the efficiency of the transcription and the mRNA translation in mammalian cells. We have also designed these own pDNA templates in order to be compatible with a scale-up and a GMP production grade, allowing you to keep the same structure from proof of concept till industrial production. As an example of our pDNA used, you can see here some quality control of our GFP mRNA showing high purity, constant size, and homogeneity. If we have a look here at the final GFP mRNA expression in an HGK cells models, we can see here that compared to the gold standard GFP mRNA from TriLink, we reach a similar protein expression. This guarantee again that our construction is as efficient as a gold standard for GMP production. For a complex sequence that are complicated to transcribe and cannot be efficiently improved by simple codon optimization, Tebubio can modify the condition of the production to increase the quality of the final mRNA. As an example, the interactions leading to secondary structure formation are usually based on a hydrogen liaison that can be limited by increasing the temperature of the transcription reaction. Playing with these parameters allows to avoid truncated form. A supplementary purification based on beads functionalized by the oligo-DT allows to isolate exclusively the mRNA with poly-A tail, eliminating all the incomplete form of mRNA. If we look here at this graph coming from TEPstation results, we can see on the left that we have two pike, one with the targeted mRNA form and one representing the truncated form. After our temperature modification on the right graph, we only observe one pike representing the unique form of mRNA that we wanted to have. Something that might be interesting for you as well is also to monitor your mRNA delivery within your models with our capacity to merge the mRNA sequence with fluotag or probes. Thanks to these techniques, we can or you can monitor yourself the transcription and translation efficiently, the release and localization within the cells, or the rate of encapsulation with its LNP solution for delivery. If we look at this picture, we have modified here the EGFP mRNA to label it with a red fluorophore. This red fluorophore allows us to see if the transcription is efficient, checking the presence of this red spot inside the cells and not outside, and also matching this green image with the GFP1 to be sure that the final protein is expressed. The yield and the quality of the mRNA with fluorophore is still very high. Here in the TEPstation, we do not see any truncated form appearing because of the ND fluor addition within the sequence. Other modifications can also be possible, like adding heavy isotope to study the final structure. The keyword here is that we will always be open to discuss your customized projects. Sorry for that. Our capacity of RNA synthesis is now around 192 RNA per batch for 100 microgram batches, producing and purifying the final mRNA in a 96 well plate. This can allow an easy and rapid identification of the best sequence with targeted yield and quality to accelerate your screening project. As I previously said, when I was talking about the different native DNA templates that we can start with, a proper template is adapted for scale-up and GMP production. At any step of your mRNA project, one of our experts can guide you from your proof of concept till GMP production. So today we have talked only about the production process, but this one is included in a complete workflow that can match the needs of every step of your mRNA project. After producing your optimized mRNA, we can propose different solutions for delivery, such as transfection reagent or LNP, and study the protein expression and its consequences in several 2D and 3D cellular models. We then propose a complete biomarker and biostatistics analysis, allowing you to have a complete set of data supporting the effect of your mRNA and the related protein expression within your model. We can help you validating every vitro steps and guide you to an optimized GMP production for your clinical phases. To conclude this presentation, I would propose these take-home messages. First, Tebubio can help you optimize and produce your mini-scale mRNA within 5 days for screening project or research-grade mRNA We can manage the synthesis up to several hundred sequences per week. Thanks to our own pDNA, we can propose you an all-in-one solution from research grade to GMP production. At Tebubio, we foster human interaction between our project expert and our clients. Do not hesitate to follow our activity and subscribe in our coming webinars to know more about our other mRNA services and platform we have to give you a complete overview of how Tebubio can help you accelerating your research.
Perfect. Thank you, Romain, for this presentation on our mRNA production services. And now it's time to move to the Q&A session. So during this live session, you can continue to ask any question you may have, and our expert will enter them. I will also be offering you a survey during this session so that you can share your feedback on this webinar with us. Okay, let's move to the first question. Okay, the first question is, what capping technologies are available in the mini-scale services?
So what we usually perform for the RNA production in Tebubio is the Cap1 enzymatic capping. But we also have a possibility to use different types of capping depending on what type of licensing restriction they have. But we are open to any type of capping as long as we have the availability for it.
Okay, thank you. A second question. Which type of RNA are you able to produce?
So basically, we can perform the in vitro transcription for any types of RNA that needs in vitro transcription to be made. And our range for the moment was from an RNA that was as small as a tRNA to as large as a SA, a self-amplifying RNA, so which basically range from a little less than 100 nucleotides to almost 9 kb.
Okay, thanks. Another question, what purification method do you offer? Oligo-DT only or HPLC as well?
So we purify the RNA using Silis columns or Silis beads. We also can offer the oligo-DT purification. We do not offer, as for now, the HPLC purification step.
Okay, thank you. Another question. The mini-scale is also available as GMP, or for the GMP production, you need an upscaling?
The mini scale is done in the lab, but it's easily upscaled to the GMP production.
Okay. What is the longest length of mRNA you can efficiently generate?
So we went as up as 9 kbs. We usually have the highest efficiency up to 6 kbs.
Thank you. Do you use, another question, do you use poly-A polymerase for the poly-A tail synthesis?
So it depends on from what plasmid we start from. If we use our proprietary plasmid from Tebubio, we have a poly-A tail that is inside the sequence of the plasmid, so we do not need an enzymatic poly-A tailing. But if we use your plasmid and your plasmid does not have the poly-A tail, we can add the poly-A tail through the poly-A polymerase.
Okay, thank you. Another one. How do you verify the length of the poly-A tail after mRNA synthesis?
We check the quality of our RNA using the tape station, which give us the length of our RNA. So if there is any anomalies with the size of your poly-A tails, we should be able to see it through the tape station step.
Okay, so many questions. Great talk and thanks for organizing it. Okay, from Rajan, first question. For screening project, having multiple mRNA, the bottleneck will still be preparing different construct of pDNA. Does the company have in-house availability of pDNA production?
Yes, we can help you make the plasmid, the pDNA production, and check with you how it will be the most efficient way to produce your your plasmid, especially if you have several sequences that are repeating between plasmids.
Okay. Second question from Rajan. Do you use in-house product for transcription or something like NEB transcription kit that shows 180-fold yield?
We have in-house products for transcription.
Any comment on purity after capping step using the bead-based purification step?
We have the same result using Silis beads than when we use Silis columns. And we never had any problems with any subsequent use of the RNA using those types of purifications.
Okay, thank you for these 3 questions. Another one, is the method of the purification that you use is sufficient for the double-strand RNA removal?
We are, I do not really have this data yet. We have not checked in depth about the double-strand RNA, but it is something that we are working on, and we should have more data to present soon, I guess.
Okay, thank you. Another question, how long is the turnaround time if your plasmid platform is not used?
It would be 5 days. So it usually takes us 5 days to make the RNA from the moment we have the plasmid.
Do you have a pathway to larger scale production? Is this a capability you have in-house?
So there is a pathway for larger scale productions, especially for the GMPs. This is by partnership with other with CDMOs, but it's not made in-house in Tebubio.
One more question about the polyethyl. What is the length of it in the pDNA, sorry, that you use?
We have a tail that is 120 A's.
Okay. Do you have any idea without UTR, those IVT mRNA be efficient enough?
We work with RNAs that have UTRs. So we have made some RNAs that without the UTRs, but it was made for our clients. So I do not have their results yet about what they think about the RNA. It will need to be tested if we really want to check how those RNA are stable in the cells, for example.
Thanks. Another question from Rajan. Have you compared quantitative results using a translational efficiency within cell or within RRL?
So I'm not sure what is RRL, but we have tested our RNAs, especially the EGFB ones, or the control ones, in cells, in different cell types. And we are, it's like what presenting in the presentation, we are quite happy with what type of result we have compared to other control from other companies.
Okay, thank you. Another question, is the poly-A interrupted?
Yes, the poly-A tail is fragmented.
Okay, thank you. Another question. Thank you for your presentation. I was wondering what is the usual turnaround time when a plasmid needs to be optimized and or produced at Tebubio?
So this will vastly depends on the size of your gene. Mostly, if it's a small gene, it could be as short as 4 to 5 weeks. If it's a very long gene, it could be up to 5 to 6 weeks to produce the RNA, to produce the plasmid.
Okay, thank you. Another question. Are there any special specification to have if we want to send you a plasmid for production?
The two most important parts that needs to be present in your plasmid is a promoter for the polymerase. We work mostly with T7, so if you have T7, it will be the best, but we also can work with other types of promoters and other type of polymerase. And the restrictions, and to make sure there is a enzymatic restriction site at the end of the RNA you want to produce. The best to work with you is to share your plasmid map so that we can work together on what we can offer and what is already present in your plasmid, so to make sure that we can use it for RNA production.
Okay, thank you. Another question. Do you offer UTR screening as well?
We do not per se by our service, RNA service, but if you are interested in, we can discuss together and check how we can construct a project together.
Thank you. That's a comment. RRR is rabbit reticulocytes lysate cell-free assay.
I see. Thank you. Thank you very much. No, we have never tested that, and that could be something we can do.
Okay, another question. Do you produce circular forms of RNA?
Yes, we do. We have a template for circular RNA.
Okay. Another question. You have talked about EV isotype for the structure. I didn't really understand. Could you explain more?
I will not be able to explain to you this as for now, but I can come back to you if you need to with more support from what we have as a document in Tebubio.
Okay. We still have time for the question, and we receive another one. Do you sequence your RNAs?
The RNA, no. We do not sequence them in-house. Our quality control for RNA are basically the concentration, the ratios that we have when we do the concentration, and the tape station step.
Okay. Tebubio offers several modified U. Could you please comment on difference between them? Any recommendation for particular application of each of them?
That could be that would be a whole webinar by itself, because it's the numbers of the UTPs that are available is extremely big, and different types of them have their different use. But basically, for what is the most common used in therapeutics, especially vaccines, is the N1 methyl pseudo U. It's because this is the one that is the highest, that has the highest expression when they are tested in different types of cells and also animals.
Okay, perfect. We still have time for a few other questions coming. Okay, what is your threshold for QC in terms of percentage of correct size?
We are usually looking for 100% of the correct size, and we usually are very close to that. The result in the tape station is basically one peak that we could check, and if it's very rare that we have different sizes, and usually our policy is that we come back to you, we discuss with you if we have any problem with this type of anomalies, and we usually try to discuss together how we can modify that and make a better work with you.
Perfect. Just to comment, thanks for the people who answered the poll. Don't hesitate to provide us your feedback to help us also to optimize the next webinar. Another question, how do you do your mRNA circularization?
To do so, we use different types of temperature changes. We follow what is basically done in the literature about circularization of the mRNA. It's basically based on the template. It's not, we do not use the same template between the linear RNA and the circularized RNA. The circularized RNA has its own template that allows us for its circularization later on.
Okay, thank you. Does the tape station pick up double-strand RNA?
It should not, because the RNA is denaturated before going through the tape station.
Thank you. Another question, how do you determine the capping efficiency and the percentage?
We do not have right now a way to determine the capping efficiency as per now, because it's extremely it's quite difficult and quite costly. But we use kits that are known to have a high capping efficiency. We are, in fact, currently working on a capping efficiency test in Tebubio to provide this new quality control for our RNAs.
Okay, thank you. Maybe a last question. We still have a few minutes. Waiting. In all the case, if you have any question popping up after this webinar in the afternoon or in the coming days, don't worry. By the this afternoon, you will receive a mail with the recording of this live session of the webinar, as well as a form to continue asking all your questions to our project manager or requesting an appointment with Celia and our experts. So, okay, 1 minute more, we can wait. Okay. No new questions, so that's the time to close this this webinar. As I told you, you will receive the recording in the afternoon. You will have the opportunity through the forms to ask all your questions or to request an appointment with our expert to discuss about your project. So we hope that this session has given you a clearer picture of our micro-scale production services, and also that we have been able to show you how Tebubio can support you to accelerate the validation of your mRNA-based project or proof of concept. So to close this webinar, and on behalf of the whole Tebubio, we would like to thank you all for attending and for all your questions, and hope to see you soon at our next webinar session. Have a good afternoon, and thank you all. Bye.
Thank you.
Bye.